the multiomics. So here I want to emphasize on something known as the thing is the same, but where you look at it, that actually changes your understanding of the thing. If you look at it very close or mid-range or far away from, you will see that this is not only a image. If I ask you what is a genome, you'll probably have one of these three things in your mind. You'll think of genome as coming out of the nucleus and the DNA contained within that, or you think of like me when I started a few years back, you'll think of genome as a code and it's a book of life that needs to be understood. Or where I stand now, I know that not only a stable sequence of code, but also these codes are expressed in a dynamic and coordinated fashion going across many different pathways. So you think of it as a network. So depending on where you are coming from and what is your interest and what is your capability of viewing, you will think of one of these things. And later on, I'll show you that the third thing can also connect to the first thing, but that will come later. So briefly, I'll try to keep on telling you in a kind of simple language what is genomics. Like everything is actually genomics. Omics means genomics, but people don't like when you say that transcriptomics is also genomics. Some kind of hardcore genomicists, they think of genomics as only this. DNA sequence variations, the set of that is genomics. So that looks at mutations, and I'll be kind of throwing in some studies on the side to show that how these such approaches has been taken. And then I'll focus on a specific case study which I've been working on. So now you see, using 2,750 cases, it could be shown that only 3 variants, they explain only a small fraction of the death due to patients with sepsis. So it's not much, but I think it's still a step in the right direction. So they say that we now need data with many more thousands of patients to have more insight. So you definitely need more than simply DNA sequence variations. Okay, so as you know, DNA is transcribed into RNA, biochemistry 101. DNA is transcribed to RNA, and RNA is basically the precursor protein. So here is a study which says that only using 21 cases, but I think it's an extremely robust study. It says that neonates who later developed EOS, they already had a differential gene expression signature on the day of birth relative to LOS, healthy neonates, or simply local infection. And you can read the numbers. They are seeing genes at the range of 1,000, like 1,000 genes, 932 up, 933 up, 977. So a lot of changes. Those of you who have been following the literature, you will know that a term called genomic storm was coined sometime back for diseases such as this. They actually coined it for critically ill patients, trauma patients, but I think that equally applies to sepsis. So there's a lot of storm-like thing, lot of perturbations are happening. So that also creates a certain kind of overwhelming complexity in the data. Similarly, proteomics has used for kind of, if it can be used, again, they used a proteomic signature for potential utility in guiding fluid strategy. And there are metabolomics for all the molecules that can be studied, or the, regulatory changes in the DNA, either DNA methylation or histone acetylation. These changes are also part of the multiomics. Now, some of you are interested in other kinds of omics like metabolomics. Some of you told me that, are you going to talk about metabolomics? I am working partly on that, but I thought for this audience, it was better to give you a sense of what can be done in a kind of actionable manner. That's why I've not presented any data that is not complete. So I'll be focusing on something. But before that, before my own work, I want to show you that this study, they integrated omics in a very interesting manner. They did both blood transcriptomics and plasma metagenomics. Metagenomics means they try to detect the bacterial genes in the plasma of adults suspected of sepsis. And they say that with this approach, they identified 99% of microbiologically confirmed sepsis cases. Actually, for this paper, it was of course highly acclaimed study, but there's a comment upon that given by one of the authors whom I read almost 30 years back, and who had actually pioneered the use of AI, then called artificial neural network, in the clinical microbiology. Now she's commenting upon that. It was very, very nice that she was asked to comment on that, on this paper. And she's saying that even after our work, within 26 hours, there has not been sufficient clinical uptake of this technology. So even if the result itself is impressive, how likely it is to be implemented in the either in the ICU or in the clinic. So there are, I have a slide in which I'll be think the question that I have in mind, which are there kind of obstacles to its implementation. Okay, so all these challenges you can think of, like sepsis is a heterogeneous disease. Is it, how do you define sepsis, or how do you define your kind of cohort, or the phenotype that you want to study? Data is complex, and multiomics data is even complex to the power 5 or 6, however omics you add. Then which platform is good for me? If I am focusing on the metabolic dysregulation, then metabolomics or lipidomics is something which I should work with. But if I approach it, whether there is immunosuppression or there is hyperinflammation or there is coagulopathy, maybe I should go for transcriptomics or something similar. After I get the result, how do I translate it into my patient? And of course, the economic considerations. Some of these issues I'll try to answer later once through my own case study. Okay. So I hope I'm not going too fast. If there's a hand, if I'm going too fast, I'll slow down. So this is another approach I would like to kind of impress upon you, that most of the time when you kind of do a study or analyze data, we take a group, very clean group of group A, reference group or control group, whatever, and then we have a group, case group or treated group. So we go to intergroup analysis. But a new mode of analysis is emerging, which is I'm calling it as a single sample analysis, in which the patient, the single patient, serves as its own reference. So because I think the hypotheses are tested, mostly tested across the groups, but the decision is implemented at the individual level by an individual doctor. So the unit of comparison for the intergroup analysis is at groups, and for single sub, of course, the individual. The type of deviation we are looking for in the first one is from one group to the other, and the type of deviation is within the same subject, how there is variation. Because of the omics scale data, you can actually see the variations within that. If you didn't have this, you could not. If you have only single data point and only blood glucose level, you could not do that. Then primary purpose of intergroup analysis is biomarker discovery, research mostly, and the primary purpose here is the clinical decision making. And the key strength of intergroup analysis is statistically robust, and a single sample analysis is clinical interpretation. So whether you are looking at the population level data or you are looking at the individual patient and trying to get the best decision for that, I have done both. I started with the intergroup analysis and then I moved to single cell sample. Okay, so this is the background. What is our approach? I talked to you about the network of the genome, and I told you that the DNA sequence is stable, but is expressed dynamically in a coordinated fashion across the pathway. This is one such pathway. Okay, so doesn't matter what pathway it is, but I have colored this pathway with each node being a gene. So it is T-cell receptor signaling pathway. So the signal starts from the left and goes towards the right, and then there are different nodes. And this coloring is by gene expression data. -1 is lowest, +1 is the highest, and you can see that each gene are at a different level of color. We can quantify this. So I quantify this, and this from all these are actual data in my set. And I look at 3 pathways: pathway A, pathway B, pathway C. And you can see that pathway look more or less green. So the score comes to -31.3. The pathway C looks more or less red, more red than green, 36.4. So we call it upregulated pathway, downregulated pathway, and a pathway B is kind of middle, not much change. So if you take the dashed line as the baseline, you can actually assign a pathway as being upregulated or downregulated. And just bringing some rigor that this is not only a visual, sort of eye candy. This actually can actually tested. So these are all rigorously tested. So the null distribution. So just to give you visually that how it's tested, the line, if it's to the right of the null distribution, it is up. To the left, it is green, and then there is a significance associated. So simply by the proportion of times, how far it is. It is too far, or it's kind of in between, or is right in the middle. That's how you test it, and that's also whether significant or not. So now, what is my hypothesis? My hypothesis stems from a specific pathway that I'm looking at, and this image is clear to all the neonatology and the pediatricians. In fact, anybody who deals with sepsis, you find DIC cases, and we believe that a pathogen induces some change in platelets, leukocyte, the tissue factor role is a thrombin generation, endothelial dysregulation, and ultimately, if there is thrombus formation, there will be DIC, disseminated intravascular coagulation. Here, I am focusing on one particular cell, leukocyte, that is neutrophil. And as you know, neutrophil extracellular trap became recently very sort of, prominent. And there are two ways that net formation can happen. But both the ways leads to, is there a pointer? Okay. Both the ways leads to net formation. These are all the DNA and histone. DNA and histone are being. How many of you have never heard of net? Neutrophil extracellular trap. Okay. So okay. Then I'll spend just 2 minutes here. So neutrophil extracellular trap is a third type of. It was actually discovered in 2004, and it is a third mechanism by which neutrophil eliminates bacteria. The first two are phagocytosis and degranulation. So in this process, what happens is that neutrophil gets some signal from its membrane, certain changes happens inside, and then two things mainly happen. One, that its chromatin becomes decondensed. It becomes sort of kind of loose, no longer condensed. One, and secondly, there is the pores are first pores are formed, and then the membrane itself breaks, and the thing comes out. When it comes out, it has also these antimicrobial peptides on its top, so on its surface. So the nets consists of DNA, histone proteins, antimicrobial peptides such as MPO and neutrophil elastase, and its main purpose is to trap the pathogens. But as a side effect, it also traps platelets. It also induces some kind of a inflammatory response, and it can cause damage to the endothelium. In short, if it is not properly controlled, it will lead to DIC. So this is a dangerous kind of double-edged sword, and we'll come back to it. We need discussion on this later. We have actually interesting discussion relevant for all of you today. Okay, so my hypothesis that is netosis upregulated in non-survivors compared to survivors. That's how I'm studying, and my data platform is completely transcriptome to start with. Okay, so you may have heard of this. So blood transcriptome studies are relatively difficult to do. They're not so easy to do because this RNA is much less stable compared to DNA. So we need to stabilize the RNA and generate mRNA of good quality. Once it is done, then we need to convert it to cDNA by process known as reverse transcription, the kind of reverse of transcription, and then you do get all the fragment. Then we get all these reads. Then we align it against the genome, the human genome, and get all this set of transcript abundance. If our genome contains 20,000, we get 20,000 values for patient. So that gene is how high it is, how low it is, all that. All the green and red colors I showed comes from this. And then we test the hypothesis. Okay. So with this work plan, what was my result? My first result was that I could test that in 16 non-survivors compared to 93 survivors, is extremely upregulated, this pathway, and netosis pathway extra upregulated with a p-value of close to 0. And this is some math, let's forget about it. So basically, I proved that netosis pathway is upregulated in non-survivors. And this is data taken at the day one of admission. And so that consists of 111 genes. You know that pathway consists of 111 genes. Actually, pathway consists of more genes, but we could not find those genes. So we found 111 genes. It's still a large number. But we know that not 111 genes are equally informative. So we need to reduce the information content, and we need to funnel that through different algorithms. One is volcano plot, the other one is lasso. These are all, lasso is a machine learning algorithm. If somebody is interested, I can discuss, but I think I should not. Should I try to discuss? Oh, fine. So basically, you can take it from me. Many papers also mention lasso. These are extremely robust methods. And from 111, I came to 24, and then I came to 9. So these 9 genes I find are the most informative in the netosis pathway, which are predicting. Now, my first prediction was 111. I have to reprove it with these 9 genes only, and we call it as a key netosis genes. We did that, but here I have switched gear with the kind of in the side in the background. From a intergroup analysis, I have now moved to single sample analysis. So basically, in each sample, right now, I'm not taking the kind of these values are not between two groups. For every subject, for every neonate, a score was generated using these 9 genes. This basically based on the rank of the 9 genes vis-a-vis other, the genes in the pathway versus genes not in the pathway. How it is changing, and you can see that in the non-separate group already it's changing too high, and it's extremely significant. And we could validate in another group. We call it validation set 1. So 9 keynet genes are they sufficiently discriminate between non-survivors and survivors at day 1 of sepsis. Okay. So what we have done, we have shown that netosis pathway as a whole discriminates between survivor and non-survivor. We have shown that out of this pathway, 9 genes are the key netosis informative genes, which can differentiate between survivor and non-survivor. And since we have used single sample analysis, we are good to kind of, we're one step toward the clinical implementation, but not yet. We have more things to do. Now, what is the relationship between netosis and coagulation? We know from literature that they are linked, but we have to prove it in this data set also. So this is a relatively kind of complex slide, but I'll tell the basic idea. We know that correlation is not causation, but correlation is a step towards the causation. In the first step, what I did is that I tried to correlate that the net score, each subject has a point, and similarly we found a coagulation score. So net score and coagulation score are positively correlated with a significant p-value. Then we showed it at that net score on day 1 can control the coagulation score on day 1 and also day 3 later on, but not the other way around. So netosis is proximal to coagulation, but coagulation is not proximal to netosis. Now, this is only in our data, that is transcriptome data. But in biology, we know that there is a bidirectional relationship because there are certain coagulation factors which can actually stimulate netosis. But we don't see that evidence happening in these patients. That's why we had to test it. Third one is mediation analysis. I'm not going to that. It is basically an analysis to prove that the data is consistent with the causal variable being netosis and not coagulation. So there is a direct effect and an indirect effect and a total effect. So using that, you can show that netosis score is actually more controls the outcome than the coagulation score, although coagulation score has some role to play. So this is that. Okay. So what this here I'm showing that 9 here onwards we're all dealing with 9 key net genes, although it doesn't say anywhere I write net score, I say key net, it means those 9 key net. So I show that these 9 key net genes, they drive coagulation. Here in this alluvial plot, it's easy to see that the red ones, the orange ones are the non-survivors. These babies, they died. The others, white ones, survived. You see that when a baby has high netosis, also likely to have high coagulation, and then the baby dies. There are a few babies who are high netosis to start with, but they don't have high coagulation, low coagulation score, but they do not survive. Then there are babies who have low netosis and a low coagulation and also do not survive, which means that there is some heterogeneity in the way the babies die. Babies do not die only by this netosis induced DIC mechanism. So there is some scope for going there, but you will find that it is much more muddy in other age groups. So neonatal age group is a little bit clean that way. Okay, so what you have done so far is that we started out with the hypothesis of netosis. We showed that netosis is associated with poor outcome. We showed that netosis is also associated with coagulopathy, and now we want to go next. Okay, 17 minutes. I have enough time. So, we are saying that netosis is a neutrophil process. Is there any evidence in our data to support that? You see, I am kind of dissecting my data in ways in which different biological questions and clinical questions can be brought up on the same data and answer those questions to the extent possible. You may or may not know, from the transcriptomic data, it is possible to calculate the cellular composition of the sample from which the data came. It's possible to do that. We have done it. And this shows that of all the cells which are present in that sample from which you got the data, the neutrophil is most highly correlated. So this is one evidence that the neutrophils are responsible for this gene expression. The second evidence is that on the right side, the neutrophil count from actual data, clinical data, and neutrophil count from the CyberSort X, that algorithm I was talking about, and there's a good correlation with significance, a small p-value. This correlation is actually much stronger in the non-survivor than in the survivors. So here there are two things I have observed. The non-survivors have a low neutrophil count, but high degree of netosis gene expression. What does it mean? It means that the netosis gene expression is leading to death of the cells, because cells are dying, and that is how the cell number is reducing. So all the data is consistent with that. Okay, so obviously my question is that because netosis has been reported in other age groups, even other conditions also, other diseases also, so how specific is my signature to neonates? Maybe it happens in all sepsis. Anybody who has sepsis, it will have this. Not so. So we took data, as you can say, 142 survivor, 22 non-survivor, pediatric sepsis and adult sepsis, and we could not. Our AUC extremely poor compared to what we got. Around 0.9 in the neonates. Why? What does it mean? Does it mean that netosis doesn't play a role in pediatric sepsis? I don't think so. I think those of you who are working with pediatric sepsis, you also do see quite a number of cases of DIC, right? If I am not wrong, you do the case of in pediatric sepsis also you do see DIC, right? So I don't think netosis doesn't happen in pediatric sepsis. But I'm thinking those 9 KINET genes may be specific to neonates. There may be other mechanisms, other pathways to death. So in the older cases, that is my interpretation up to now. Okay, so then we have to generate some data from here, from India, our own data also, some data other than what we have already seen. And here I'll just draw your attention to the left side. In this data, it's a small sample generated from Ames and Cincinnati. Only 14, 8 survivors, 6 non-survivors. What you see, and we are using the same 9 KINETs which you got from the other. So it's a kind of independent validation of that net score. You see, only using the best clinical parameters I could get from this. And those best clinical parameters was taken from a set of babies which are not here, from another 100 something babies. And we are seeing we're not getting a good AUC. But only using net score on the same babies, we are getting it close to not 0.9 yet, but 0.85. And when you combine both, this could not be sufficiently improved and it is statistically insignificant. So here we can say that if you have a baby and you are having all the best clinical parameters, if you add the net score, is it going to improve your predictive ability for non-survival? I think yes. Okay, we also wanted to see if in this baby DIC information was available for some, like we had 3 babies with DIC. We saw a trend with net score, but we could not prove it statistically. So this much we have achieved so far. Okay, so with this, what I have done is that I proposed first that netosis is a predictor of poor outcome. I found that the pathway as a whole is a predictor. then out of that 9 genes could be refined. They are probably the best set which can predict, and we could prove that in a small Indian cohort also. Now here I want to bring in something which is very relevant to you and which is a sort of is a kind of debate going on in the field. So there is a school of thought that neonatal neutrophils are not sufficiently developed to cause netosis. That means they are impaired in netosis. This was probably from 2009. First, when it was said that neonatal neutrophils do not readily form from nets, and so in adults, as you can see here, in adults with a given LPA, there is a robust response, but the neonates both in term and preterm, there is a low response. Another group also showed that in adults, both at 2 hours and 3 hours, good response, but newborn take more time to give the response. So whether they do not give or they give less response, this was a thought process which started then, and it was thought that netosis is not relevant to the sepsis process. Then in 2016, it was proven that there are endogenous regulatory peptides known as NNIF and similar, which are produced by the placenta and which actually reduce netosis, can reduce the ability of neutrophils to mount netosis in the intrauterine phase. And so this placental protein was told to be a serine protease and which works on the alpha-1 antitrypsin and breaks into smaller fragments in 2021. So it was no wonder that in this paper in 2020, when they tried to use cord blood to predict if the baby will have, those babies who have EOS, I think they went for EOS. EOS, LOS, sepsis, and try to say that if the cord blood has netosis markers which can predict sepsis, they could not find. So they also acknowledge that it is possible that those endogenous peptides are actually inhibiting, but they didn't go strongly to say that. But I think they kind of diluted their own study a little bit. I believe that cord blood is not the right source of a right tissue to study if a sepsis is associated with netosis. You need to study the neonatal blood because the action is happening after birth. So this is my thoughts. You may or may not agree. I mean, this requires, of course, a more debate than we can accommodate in this forum. But in the intrauterine life, there is a strong inhibition of netosis by placental factors, placental enzyme, which produces NNIF and which prevents netosis. So also because placenta is supposed to be a sterile space, so this is okay. And netosis also can be problematic because it can, as you have seen, that it is a kind of double-edged sword. But after birth, once the NNIF level starts falling, we know that by 2 weeks they're gone. But I think much before that also they start falling. Then the net formation becomes possible. So babies at risk of infection, they're actually protected by the additional mechanism, but also paradoxically, they're also at the risk of net induced PI. So this is how I'm kind of sort of reconciling the multiple observations, and I still think that for the neonates, net study has to be done in a manner, in a sort of balanced manner, so that we know that NNIF effect is gone and the netosis is starting, but it is not yet so well controlled. Because net, as you know, it needs to be controlled. It also needs to be removed quickly by DNase, by DNA which is produced. So if it is not reduced, not removed quickly, then it will cause all the DIC and other things. Okay. So what is my next step? My next step is I am proposing to you that this is what I want to do. And see, I propose in multiple scientific audiences, but I would propose to only pediatricians and neonatologists. You can tell me whether it is feasible. So my multiomics proposal consists of 2 things. One is pathogen genomics and one is transcriptomics. Both can be done by machines which are now, let us say, maximal this size. Okay. So you have all these clinical biochemistry, clinical microbiology labs. If they have a clean enough and they have a basic molecular biology setup, now with COVID, every lab has that. With this only, we can have everything. We can have the sequencer, we can have the computer, we can have everything here. GPU server comes along with that. So it's a very small machine, footprint is less. So both pathogen DNA sequencing and transcriptomic sequencing can be done on that machine. And I would like to have a diagnostic arm. The first one is the diagnostic arm. I told you about the 99% accuracy they could do with host pathogen integration. It is the same line, and a prognostic arm. Both can be incorporated into a single sample, 1 ml of blood coming out from the baby. And we have started some simulation because I was thinking, being in a research institute, what can I do? So I started simulating. I started taking healthy neutrophils and stimulated with the pathogens which you already knew that it is infecting the babies, because those pathogens were isolated. We have another group for pathogen sequencing. So we get pathogens. We are now focusing on Klebsiella pneumoniae. So take healthy neutrophil, stimulate it with Klebsiella pneumoniae, and see that. After that, we try to remove the human DNA and take only the other DNA and see that by sequencing how fast you can get the result. So far, our record has been about 10 hours. So I think if you give the antibiotic twice a day before your next antibiotic is due, this report should be available. So that is my simulation, and I think it is a feasible proposition. But there are practical questions. We are coming almost to the end. So, but here I really want to ask you these questions. I have already thought for you, and you also will be having these thoughts in your mind. That do we really need a sequencer in the NICU? Do we really need it? Is it not too expensive per neonate? Can our lab staff handle this technology? Is this technology for research or for clinical care? Then what about regulatory approval? Is it approved? Then will clinicians need to interpret the results? Then who will want the results come? Who will act? Or the data will be just like there? Somewhere it will be there. It is a good, looks very nice and looks very flashy, but it is just there. There is no action. So all these questions, and some more questions also are there, but these came directly on my mind. I will try to convince you that most of the answer to most of the questions are positive. Let me go one by one. This I am telling from my own experience for the last few years, more than a decade. I've been thinking about this and working on this. Do you really need a sequencer in the INICU? Depends upon you. If you are little, adventurous, you need. Okay, but if you are saying no, I'll just outsource. My plate is full. I'm already sufficiently busy. I can outsource. There are already competent people doing it. You can take that. There are already companies which are already doing it. That is not even our institute. Also, you can use. You can send us up, and we'll do it for you. But if you want to keep it, I'll tell you that it is feasible. It's feasible and it's not that expensive. The machine itself is on that. I'll come to per sample cost. I'll come to. Is it too expensive for you? Yes, it's expensive. 10 to 15,000 is the current cost for one sample. Now, this cost has to be, I'm not an economist, nor have I thought of it in a commercial point of view, but this has to be thought of in context. A neonate, if even NICU spends between 30 to 50,000 per day. Can you reduce the stay of day by a targeted, selection of the baby, by screening of the baby before it goes for sequencing? Can you reduce the cost? Somebody needs to do the analysis. And I think that is the analysis properly done, because many babies who don't need antibiotic, they do get antibiotic. We know that. So the answer probably to me, it will ultimately be less expensive, and it will actually reduce cost. Can our lab staff handle this? Well, the kind of interfaces for that are developing every day. Even we are working on it. You need some help. That staff needs to be trained. But I think that is true for every machine. Any new machine comes, the staff needs to be trained. Is this for research or for care? Both needs exist. Okay, if you are into research, then also you'll find the omics technology extremely useful. But I am given an alternative way to think, that you can also think for clinical care, but may not immediately, maybe after 4 or 5 years, when some company will come to you that this system exists, can you do it for us? Now, regulatory approval is actually the need is less here because you are already doing genetic for the rare genetic disorders. You send neonatal blood uh, for sequencing, and here the ethical risks are actually less because you are not identifying the person. You are identifying the pathogen and you are identifying the host response, which are not so identifiable as compared to the DNA sequence. So actually regulatory needs are the current regulatory approval, whatever we have, that can be used for this. Will clinician need to interpret omics? No. The way we I should have put on a report here. With the way we put a report is that only for pathogen component, I had a report prepared. Our algorithm gives a report. This is a sample. This is the most probable pathogen. 99% with some 99%, 90%, 95% confidence. How much we say that this pathogen, further clinical correlation is needed. That will be the report, and then the clinician can work on it. Think that if that looks like a possible organism, and there are many what should I say, and also we did do that. If you start doing it, you can have a pilot project. You will probably get a grant from BIRAC or ICMR for this, and then you can do 50 cases to sequencing as well as microbial culture. How many times the sequencing is missing the microbial result, the gold standard result, or the clinician agrees with it. Okay. Not many times it will miss. If anything, it will probably find some culture negative cases also. Yeah, and who will act on the results? So this is a long term. So I am envisioning like this. Once the omics in general becomes a part of the clinical laboratory ecosystem, so automatically an alert will be generated. So once, let's say, net score looks promising, automatically an alert will be generated. Somebody, because these babies are around the under care around the clock, so that alert will go somewhere, and whatever the existing needs standard. So these are long-term questions, but definitely the actionability of the result is something which can be put into our clinical workflow. Okay, so this is kind of in my support. So I know I've been talking a little futuristic. Well, I have been like that. So I started learning programming. So this may be my own approach. But I believe that much of it is already here, and you all will be using it, this technology, in a few days. It's no longer only researchers. I mean, research component is there, and also for your own clinical use, you need to learn about these things. So I really liked, I talked to ma'am also, Professor Uma Maheshwari, that project on ChatGPT. I think in extremely valuable such projects we need to do, and these results should be, sort of communicated time to time and see. Only one suggestion is that ChatGPT's version changes. So within 6 months, they keep on changing news. That also was raised. So it's a really question for you whether you want to change the version or keep the same version, because if you keep the same version, your result will be less efficient than what would have happened if you had taken the new version. If the new version keep on changing, then your study will no longer be rigorous for this. So that is a kind of catch-22 situation. But thank you for doing that. With this, I'll come to the end of my talk. So I've been generously given funds to do my research by DBT, ICMR, also by NIBNG. Right from the very beginning, my work has been supported. I've collaborated with multiple hospitals, and I'm grateful for their support. And Dr. Samshiti Bhattacharya, I needed to mention because he's a statistician. I myself not a statistician, although I know a lot about data analysis, but still I've taken help from him in understanding and sometimes putting in context, sometimes slowing down, also not doing things. And my students, I'm extremely grateful for my students. These two students, Deepshika and Manisha, are working on these projects, and someone who has already passed what was the adult sepsis component, which I did earlier. So with this, I count my close, and I thank you very much, and I've taken the first step.
Thank you, sir, for the wonderful talk. It is open for discussion. I think the talk is very interesting and stimulating, but I am not very sure whether in the neonatal NeoCORE, whether they will be able to handle unless there is a particular person, as you said, who will monitor the equipment, and maybe the microbiology department in the institution, for one machine in the institution where the sample can be sent and then definitely it will be useful because you are able to predict the bacteria, mortality, all these things. It will be good. I think it has got a bright future.
Oh,
That's why.
okay.
It's like liberating from a tangled net.
And thank you for the wonderful